Here on the H&E staining of the IDC, we can see that all the nuclei are stained purple. Along with it, all the epithelial cells are interspersed with the stromal tissue, confirming carcinoma invasion. There's no differentiation between myoepithelial and epithelial cells. The ducts, as you can see, are not distinct from the stroma itself, apart from low quality of collagen seen within the stromal tissue. The patient shouldn't have many lobules nor ducts within her breast, as she is postmenopausal. Since the postmenopausal breasts have a reduction in glandular tissue, this has relatively increased the stroma, which is not seen in this core biopsy. When magnified closely, you can see in figure 2 that we have pleomorphic nuclei. These are nuclei which is different in shape, as well as prominent enlarged nuclei, which is known as the carcinogenous cells. During IDC, we can see that mitosis happens within the cells where they are split. As in figure 3, we can see that the tumor inflates all the adipose tissues, i.e. the fat cells, proving that this could be an IDC carcinoma. On the slide here for estrogen staining, we can see that the pleomorphic nuclei are stained in brown intensity colors, confirming that the presence of estrogen receptors expressed in the tumor cells. The higher the intensity, the greater the ER expressed. We can see that there's a greater distribution around this area that the cells are with a high expression of ER. Estrogen alters the metabolism of carcinogenic substances, stimulate their cellular proliferation, and induce receptor protein synthesis and DNA synthesis as well, which can stimulate the development of tumors if uncontrolled. In this biopsy is the progesterone staining, and this is very similar to the estrogen one. Here you can also see cells that are a very high degree of PR receptors expressing within their nuclei. This is directly linked to the genes involved in overexpression of the PR receptors. What we need to know here is that the PR and ER receptors are normally associated with invasive ductal carcinoma of the breast. The biopsy here is for HER2 receptor staining. The HER2 is a transmembrane receptor and can easily be found expressed solely on the epithelial cells of the carcinoma. This can be shown around here, here, and here, any place where there's a dark patch of line. There's no positive reaction seen in the stromal collagenous tissue, but just the cells associated with the invasive tumor. This confirms that the carcinoma originated from the epithelium of the ducts. Here, both keratin 5 and keratin 14 antigens are stained negative by its monoclonal antibodies using the immunochemistry chemical method. These are normally present in the myoepithelial cells in the ducts and lobules. However, in both these slides, you can see that there's hyperplasia within the lactiferous ducts, as shown, of the epithelial cells. This is seen as the epithelial cells have punctured through the myoepithelial layer and the basement membrane covering the ducts have invaded the surrounding stroma tissues. This is another way of determining whether the cancer is linked to the myoepithelial or epithelial of the ducts or lobules of the breast. Here, the SMM is very similar to the keratin, where it has stained all the tumor cells negative, though it has expressed the myoepithelial cells around the duct, as well as blood vessels. This is to show that hyperplasia is still happening, and is another way to show that this is negative for the carcinoma.